Good morning, students. Today we learn about transdermal drug delivery systems. Myself, Dr. Kunal Patel, Professor, Sri Swaminarayan Sanskar Pharmacy College, Jhunda. Learning outcome of transdermal drug delivery system is, after learning of this, you can learn about what is TDDS, benefits and limitation of transdermal drug delivery systems, structure of human skin, basic requirements of transdermal delivery systems, types of transdermal delivery systems, manufacturing process, and testing of transdermal drug delivery systems. What is transdermal drug delivery systems? Transdermal drug delivery systems are adhesive drug-containing device of defined surface area that delivers a predetermined amount of drug to the surface of skin at a program rate to reach the systemic circulation. Benefits of transdermal drug delivery systems. Therapeutic agent means drug delivered at a controlled rate through the skin into systemic circulation. Maintain plasma level of drug from 1 to 7 days. Controlled delivery resulting in more reliable and predictable blood levels. Improved absolute bioavailability. More convenient and improved patient compliance and convenience. Increasing efficacy and typically decreasing the toxicity level of drugs. Limitation of transdermal drug delivery systems. Drug must have some desirable physico-chemical properties of drug so that they can easily penetrate to the stratum corneum. Amount of drug doses should not be more than 20 mg per day. Otherwise, it is very much difficult to transfer from one layer to another layer. Skin irritation or contact dermatitis is another disadvantages of transdermal drug delivery systems. Clinical need is another important area. Barrier function of the skin changes from one side to another side on the same person, from person to person and with age. Here we see the structure of human skin. Basically, there are three layers: epidermis, dermis, and subcutaneous tissues. The drug applied on the skin passes through these layers and goes into the systemic circulation. Now, think about what are the basic requirements to prepare and evaluate transdermal drug delivery systems. Let's see the basic components of transdermal drug delivery systems. The primary requirement is drug, active pharmaceutical ingredient. There are some physical chemical properties of drugs are important for the selection of the TDDS. Polymer matrix. The polymers are of natural polymers, semi-synthetic polymers, synthetic polymers are used to prepare the transdermal drug delivery systems. Permeation enhancers that are help to increase the permeation of drug through the skin layers. Adhesives are the solutions. They are used to increase the adhesion of the skin to the patches or transdermal drug delivery systems. Backing membrane and release liner are also another requirements of this TDDS. Now see the different types of transdermal drug delivery systems. Basically, there are four types of transdermal drug delivery systems. First one is single layer drug in adhesives. Here, the drug is added into this adhesive solution and this solution is applied on the backing membrane and between the liner and backing membranes. Second type is multi-layer drug in adhesive systems. Here, it is similar to the single layer drug in adhesive systems, but there are two layers of drug in adhesives and these two layers are separated by the membranes, and the other requirements is backing and release liner are similar to the single layer drug in adhesive systems. Next one is drug reservoir in adhesives. Here the drug in the solution or suspension liquid formulation is placed between the backing and the membrane. Then the adhesive layer is placed, and at last the release liner is applied to the adhesive layer. And last one is drug matrix in adhesive solution. Here the semi-solid matrix of drug is placed between the adhesive and the release liner, and they are applied to the skin structure for the transfer of drug to the systemic circulation. Now we see the manufacturing process of the transdermal drug delivery systems. First of all, the drug and adhesive solutions are prepared by adding the drug into the adhesive solution. Then these drug adhesive solution are applied on the backing membrane, and then they are dried using the suitable equipments. After drying, this backing membrane containing the drug adhesive solution is laminated using the release liner, that is known as a film lamination. Then this laminated film containing the drug adhesive solution, backing membrane and release liner is cut into the suitable size by using the die cutters. They are packed in a suitable packing systems, and then the final product is ready for the dispatch. This is the equipment used for the application of the drug adhesive solution on the backing membrane, and then the drying of the solution with the backing membrane is carried out using this machine. It is generally manufactured in the Switzerland. This is the equipment used for the lamination of the backing membrane and the release liner. This is the equipment used for the cutting of the patches into suitable size. And at last we will learn about the testing of transdermal drug delivery systems. This is important part of the transdermal systems. So it includes different tests. First one is chemical test, that includes the content of drug present in the patch, content uniformity, purity, and the residual solvent present into the transdermal patches. Second test is physical test. This includes the release of the drug from the transdermal patch, and it is characterized by using the USP apparatus 5, 6 and 7. Another is test for adhesion. As you know that the transdermal patch remain adhere on the skin of the surface. So this adhesion of the transdermal patch is determined by rolling ball tack test, prop tack test, field adhesion test and shear test. Continuous toxicity. In that case, contact dermatitis, growth of microorganisms, cytotoxicity, sensitization test is carried out. Percutaneous absorption model, that include in vitro testing, in vivo testing. In that cases, the France diffusion cell is also used to check the permeation of the drug through the skin layers. Thank you for patience listening.